Good afternoon. My name is Joanna Korecka and I'd like to introduce you to our project entitled Dentatorubral-pallidoluysian atrophy, DRPLA, development of an ASO therapy and understanding the impact of ATN1 CAG expansion. DRPLA is a rare genetic neurodegenerative disorder with widespread central nervous pathology caused by an increase in number of CAG trinucleotide repeats within the atrophin-1 gene, ATN1. Anything above the 48 CAG repeats will induce clinical onset of the disease. There are no disease-modifying treatments available at this moment. Antisense oligonucleotide-based gene therapy has proven promising for neurodegenerative diseases already clinically approved for SMA and a subform of Batten's disease, and currently being tested for HD, ALS, and other neurologic diseases. The aim of our project is patient-specific therapeutic development. Our patient is Tayson. Tayson is 14 years old and shown an early onset of DRPLA at 4 years of age. Tayson has 67 CAG repeats in his ATN1 gene. We've gained access to Tayson's fibroblasts through a skin biopsy and reprogrammed them to generate iPS cells. Through a number of neuronal differentiation protocols, gene correction, or a knockdown of the pathologic allele, we're currently developing neuronal cellular models to study this disease. By comparing the patient-derived neurons to genetically generated control cells, we want to discover pathologic phenotypes of DRPLA. Furthermore, by testing ASOs for knockdown of these pathologic allele, and we aim to reverse the disease phenotypes. Our final goal is to develop treatment for Tayson. Our ASO design strategy is based on the fact that CAG expansion induces AT1 protein aggregation in the brain. By gapmer ASO design, we initiate the intrinsic RNase H activity and degradation of the pre-messenger RNA, therefore decreasing the AT1 messenger RNA and hopefully changing the cellular phenotypes. We had performed a screening of multiple ASO candidates in human neuroblastoma cells BEM17 and found that many of these candidates showed a decrease of AT1 messenger RNA, but 2 of them caught our attention. We had therefore introduced these 2 ASOs into PS-derived human neurons and found a very efficient AT1 messenger RNA degradation by 80% compared to naive, control, and Huntington ASO design. But how does AT1 loss of function affect cellular survival, growth, and health? Not much is known about this in the literature. Therefore, we had designed our own experiment by comparing the AT1 decrease in 4 stages of neuronal development. We will look at human iPS cells, neuroprecursor cells, iPS-derived neurons, and even human neurospheres, which contain multiple cells from central nervous system. The main goal of these studies is to assess the ASO efficacy and safety as a therapeutic strategy.